The goal of this experimental protocol is to investigate the neural correlates of immediate and long-term impact of explicit and implicit emotional control using functional magnetic resonance imaging. This protocol involves assessments of emotional ratings of pictures and of the memory for these pictures as a result of inducing the goal to regulate emotion. Recording of brain imaging data is performed during the rating task. The goal to control emotions is induced either explicitly by instructing the participants to control their emotions, or implicitly by priming the participants with emotion control words embedded in a so-called scrambled sentence task. Analysis of brain imaging data targets activity in regions associated with basic emotion processing, such as the amygdala, and brain regions involved in emotion control, such as the prefrontal cortex, particularly lateral and medial regions.
The main advantage of this technique over existing methods, like those mainly focusing on the immediate impact of emotion regulation and typically targeting deliberate emotion regulation, is that it allows assessments of both immediate and long-term impact of emotion regulation, as well as comparisons of automatic and deliberate emotion regulation induction.
This method can help answer questions about opposing emotional biases, such as those observed in healthy aging, where there is a positivity bias, and depression, where there is a negativity bias, which seem to be associated with enhanced ability to automatically engage emotion regulation strategies observed in healthy aging, as opposed to impaired ability to engage emotion regulation strategies.